Okay, hi guys. Welcome to, let me put these up for a second. Welcome to a video about how to crunch data and just sort of like what data is and how to use it. You're gonna want, while you watch this video, to have your OneNote guide pulled up on your computer, because we're gonna fill it in together, just not in person this time. You guys are gonna watch the video and fill it in while you're watching. So I would recommend maybe even watching this video on your phone so that you can have your OneNote on full screen, or if you're at home and you have two different computers, you could watch on one and fill in OneNote on your school computer. But you definitely want to be able to fill in your OneNote and watch at the same time. So go ahead, and if you need to pause and take a second to get that figured out, go for it. And then when you're done, unpause and welcome back. So we'll talk today about data and how it works. I'm going to start just by kind of talking about what data actually is. And that is anything that's observed and recorded during your experiment. So before we go any further, we need to take a second to talk about the differences between making observations and making conclusions. So observations are just things that either you measure or you notice that are super objective. And objective means that whether it's you or me or some other random person, we would all come to the same statement about what's happening. So it's no opinions. It's just, I'll say objectively. I want to say observing because it's in there, but objectively recording changes or qualities of what you're testing. So your observations could be related to size as long as they are measurements. So if you said this plant is 12 inches, that is a good observation in science. If you said this plant is tall, that's an opinion. That's you and I might not say the same thing about whether that plant is tall. So that would be more of a conclusion that you would need to support with your data or with your observations, which would be the actual like number measurement of your plant. So when you're doing this, just think about, would someone else say the exact same thing, or would someone else be able to argue with me about whether this is true? And if not, then it's probably an observation if they could not argue with you about it. All right. Conclusions are subjective statements supported by your data. So once you had measured your plant and you knew that your plant was 12 inches and you looked around and you saw that all the other plants that you were measuring were 5 inches, then you could say, in conclusion, this plant is tall because compared to all of these other plants, it's has several inches more of height. And then you could go into, whatever you did to that plant that presumably made it so much taller than the others. So that's the difference between observations and conclusions. All right, so going back to just talking about data, your data can either be quantitative or it can be qualitative. And we'll talk about the difference right now. So quantitative data, when you hear that word, it sounds like quantity, and that's helpful because it's related to counting. So quantitative data is usually going to be number data. It's going to be related to, move myself over here. It's going to be related to measurements. And it's always going to have units. So it will be things like inches. In science, we usually use centimeters, meters, temperature, weight, or mass, or just, the number of, when we do our experiment this week, it'll be the number of ants that show up to your food choices. So usually if it includes a number, it's probably a quantitative data measurement. And that's most typically what you'll see in tables and graphs is quantitative data. But qualitative data is sometimes just as important. So when you see the word qualitative, think of qualities. And that's what it is. It's describing in words what's happening. So qualitative data could be things like color. It could be things like texture, shape. Qualitative data is going to be really important later in the year when we actually grow bacteria and we are looking at the shape of the colonies that are growing, the colors of them, textures of them. It's really important for things like that you can't quite measure with numbers, or you can, but it's helpful to also talk about what you're seeing because it helps to identify the different species that you're looking at or something like that. Similarly, when we look at microscope data, we will think about qualitative data as well. So sometimes your data is literally drawing a picture of what you're seeing under the microscope, and that would be qualitative and not quantitative. So just remember when you are thinking about these words that this is quantity, numbers, and qualitative is going to relate to qualities. So that should help you out. Okay, now come down here and there's a little place for you to try identifying whether things are qualitative or quantitative. So take a second, pause me, read these, and answer. Ready? Go. Welcome back. We're going to go over these answers together. So if you had a plant that grew 3 centimeters, 3 centimeters is a quantity, it's a number. That's going to be quantitative data. For the second one, the temperature of the water became several degrees warmer. Several degrees, even though it didn't give you a specific number, several degrees is a number. So we don't know what it was in this situation, but it was a number. So this would be quantitative as well. And then finally, the plant kept by the window had lighter green leaves than the one kept on the desk. This is related to color, so it's a quality. Qualitative. So all of these things are useful as data. They're just different types. So we'll practice more with this later on, but once you get the hang of it, it's pretty easy. Okay. So now the other thing I want to talk about in this video is how to analyze your data once you've collected it. So typically in science, when we're analyzing data, we are going to use tables and graphs, which sometimes graphs are called charts, especially if you use like Google Sheets or Excel to make your chart. It will be called chart. It's graph. Tables are always going to be the ones with the boxes in the columns like Excel or Google Sheets look. Okay, so when you have a data table, you should always make sure that your tables have a title. Up at the top, you want whoever's reading your report to be able to figure out what your data is showing. So you want to make sure you have a title up there. And then you also want to make sure that you include units. So make sure you're telling us, what are we looking at here? If it just says temperature, we don't know what the unit was. So we don't know if it's Celsius, Fahrenheit, Kelvin. Same thing with length. We don't know if we're talking about miles or if we're talking about centimeters. So it's really important to make sure that you include the units in the title of your different columns, which I'll show you one in a second. And then your graphs should always have a title. Again, we're going to want to make sure that we know what we're looking at here. And then you also want to make sure that you have a y-axis label and an x-axis label. The other thing that's really important is that your axes are numbered with even spacing. So whatever your sort of interval is going to be as you go up the axis or go across the axis, you want to make sure it stays the same. So if you're going to count by threes, you count by threes the whole time. You always put the same number of boxes in between your different intervals. So you can't be like, all right, I'm going to count by ones and then I'm going to switch over and I'm going to start counting by tens. You can't do that because it ends up making your graph really hard to interpret and read and can actually give people the wrong impression of the results of your experiment, which happens sometimes even if you are just watching the news and you see graphs, sometimes they have these really weird axes. So you have to pay close attention because sometimes they're not actually showing you the data that you think it's showing you because of the way the axes are set up. So just as a reminder, before we go any further, I'm going to go over here and just draw a tiny little graph that this is the y-axis going up and down, and this is the x-axis going across. And you would want to, of course, label these for whatever they were. Normally on your x-axis is your independent variable. And on your y-axis is your dependent variable. And then, of course, up here you would have a title. And your graph would go in the center. Okay, so now let's just talk about different types of graphs that we're going to be using this year in here. These are the most common ones. There are, of course, other types of graphs as well, but these are the ones we're going to be using most often in biology. So the first one is a line graph. Line graphs are going to be most useful for data that involves time. Or rate. So the rate of growth, anything that involves a time measurement is probably going to be best shown in a line graph. And those are the ones, we'll just put a little one over here, that this would be, line graph is the one that has the points, and then we connect the dots to show what's happening. This is a line graph. Bar graphs are going to be most useful for comparing different trials or groups. This is what we're going to use for our ant experiment because we're going to have 6 different foods. We're going to have a bar graph to show how many ants came to the different food types. It'll be really nice for comparing those 6 different food groups and seeing which bar is the highest. So this is the bar graph, looks like this. And then finally, you have a scatter plot, which is used to helps find correlations between 2 variables. We aren't going to be using scatter plots super often, but you will get to check out some this week in class when we use this data crunching website called CODAP. So what they look like is you just have your axes and then you have your points, but they aren't connected by a line this time. They're just free points. And then what the scientists can actually do is they can look at the scatter plot and draw a line of best fit, which we might have done in math class before, but in this one, it probably would be like about here, which shows sort of like as X is increasing, Y is also increasing, whatever the variables would be. It would show, in this one, it would show that they are correlated. Some scatter plots would just have dots all over the place, which would show you like they're maybe not connected to each other. All right, so now we're gonna have another chance for you to try it out. You're gonna come down here to this try it section. We're gonna use this data here in table 1 to make a graph over here on the graph paper. So the first thing you should do when you do this is figure out what type of graph you should use, draw your graph, and then restart the video. So go ahead and graph now. So we're going to come back and make this graph together. Because the graph is related to time passing in minutes, I'm going to use a line graph. Hopefully you used a line graph too. And I'm going to label my axis down at the bottom by counting by 5s because that's what happened in the chart. So I'm going to do every 2 squares. If you did every 1 square, that's also fine. 10, 15, 20, 25, 30. Normally, I really like when my students fill up the entire graph section. So I didn't do it on this one, but if you're graphing, try to expand all the way out and use your entire space, just because it makes your graph look a lot nicer and neater and easier to read, as opposed to like crunching things into really small areas. Okay, and then my dependent variable was the number of ants that came to the dropped chip. So I'm going to count for this one also by 5s. Yeah, also by 5s. I'll go every 2. So 5, 10, 15, 20, 25, 30. I wish that this was a little bit taller, but it's not. So that's okay. All right. So now I've got my graph drawn. I need to label my axes. So my axis down here on the X is minutes. And my axis up on the Y is number of ants. And I'm going to make my points. So after 1 minute, which I'm going to put down here, like almost to 0, there were 0 ants. At 5 minutes, there were 0 ants. At 10 minutes, there were 3 ants, so kind of right about here. At 15 minutes, 12 ants, so right about there. 20 minutes is 26 ants. And, oops, wrong spot. Should have been at 20 is 26. 25 is 24. And then 30 is 31. Okay. So then the next thing you need to make sure you do is connect your dots. If you are doing a graph that has two different lines, so maybe you did two different trials of this, or you did it on two different days, or you did it with a chip and then another food, you would want to make sure that you use either a different color line, or one line would be dotted and one line would be solid, just a way to tell the difference. So now here's my almost finished line graph. The only thing it needs now is a title. So I'll call it the same thing as my table: ant feeding over time. Okay, so once you've finished your graph, you come down here and it's time to draw a conclusion from your graph. So take a second and answer these two questions, pause the video and restart it when you are done. All right, so there are many possible correct answers for either of these questions. As long as you have made a statement and supported it using your graph or your table, then that is correct. I'm gonna say for mine, after some leaving the food source. And I'm gonna say that my graph supports that because the data for minute 25 was lower than the data point for minute 20. So now this doesn't mean that this is 100% completely the right answer, because if you think about it, there could have been ants leaving the entire time, just more ants coming. So you would need more tests to confirm for sure that this conclusion was accurate, but it's a good answer to this question because the data supports it, and that's all you can do. So cool. I hope you guys learned a little bit about data crunching today. We're going to be working on this week and also crunching our own data once we get into next week for our ant experiment. So I'll see you in class at some point, either on Zoom or in person. And I hope you have a great week. Bye.